I'm Dan Kastner. I'm the Scientific Director of NHGRI, for anyone who doesn't know me. And it is my absolutely enormous pleasure to have the opportunity to introduce to you today's Wednesday afternoon lecture series speaker, Dr. Evan Eichler, who is a professor of genome sciences and also a Howard Hughes Medical Institute investigator at the University of Washington, and is really one of the pioneering leaders in the field of human structural DNA variants and their effects on human disease. Dr. Eichler was born actually in Cheyenne, Wyoming, and then spent his early years of his life in Canada, and actually got his undergraduate degree in an honors program in biology at the University of Saskatchewan in Saskatoon, Saskatchewan. And then he went to another part of the Wild West, namely Houston, where he did his graduate work in human molecular genetics with David Nelson, and there really started to work in earnest on this question of DNA instability and mechanisms that regulate triplet repeat variability. He then went on to do some postdoctoral training at the Lawrence Livermore Labs, and then from 1997 to 2004 was on the faculty at Case Western, and then in 2004 took his current position at the University of Washington. During the course of his career, he's really established himself as one of the world's leading experts in structural and in particular copy number variation in evolution and in the human genome. He's the recipient of a number of different prestigious awards over the course of his career, and just naming three of them. He was the recipient of the Basil O'Connor Young Investigator Award from the March of Dimes Foundation, the recipient of the Kurt Stern Award from the American Society of Human Genetics, and he gave the Mendel lecture of the European Society of Human Genetics a year or so ago. In 2012, he was elected as a member of the National Academy of Sciences. In any case, it's really just an absolute treat to have the opportunity to have him here today. And the title of his talk is going to be Autism, New Mutations, Genes, and Pathways. Dr. Eichler.
So thanks, Dan, for that introduction. It's a pleasure to be here with you today and to share with you some of the most recent data that my lab and others over the last, I would say, 5 or 6 years, but with a specific focus in the last couple years, have learned about the genetics of autism. So I've approached this question really from a slightly different perspective. I see autism primarily as a disease of development. Neurodevelopmental disorder, which has historically been thought of as essentially impairments in social interaction, behavior, repetitive behavior, and language. If you believe the CDC estimates, the current estimates, I think as of April 2014, is that on order of 1 of every 68 boys born in the world today suffer or are on autism spectrum disorder. That number is about half for girls. It's about 1 every 189 births. More broadly defined developmental delay, which includes things outside of the autism spectrum, but particularly inability for children to meet certain milestones, can afflict as much as 2 to 3% of the general population. One of the interesting aspects of this disease, which we have not completely resolved, maybe we never will, is the gender bias. We know that for diseases such as developmental delay and intellectual disability, that males are 1.4 to every female that's been identified with the disease. In autism, at least the samples that we have actually particularly studied, the male bias is 4 to 1. And for high-functioning autism, previously known as Asperger's syndrome, the disease has a male bias of about 6 to 1. Now, despite the potential heterogeneity of this disease in terms of its etiology, the genetics of autism has been clear for over 40 years. We know there's a strong genetic component. The current estimates of heritability place it up there as about 50% or more in terms of, and this comes primarily from family studies and identical twin studies. A lot of work was done with identical twins to actually estimate concordance. So the estimates of concordance for monozygotic twins is 70 to 95%, and for fraternal twins, it's 5 to 20%. So if the background rate is 1%, there's obviously a significant inherited component as well running in at least a subset of the families. In the mid-2000s, searches to actually identify common genetic variants as a cause or potentially source of autism really came up with relatively few loci that could be replicated. And there's been arguments that, in fact, much larger sample sizes are needed to make this happen. We knew from studies of diseases like Fragile X syndrome, diseases such as Rett syndrome, that monogenic forms caused by a single gene that was tracking within families might account for as much as 8 to 10%. So the question really facing the field, I would say, of autism genetics circa 2005 is, where's the rest? And I would say that a particularly important breakthrough in this area came from the study of large copy number variations. So large copy number variants are large-scale deletions or duplications that exist within genomes. For the most part, they're individually rare but collectively common. And it was found in a series of studies, starting first with studying of children with developmental delay from Kulin DeVries' group, some work done by our group looking at specifically regions of the genome prone to recurrently delete and duplicate, and later work from Mike Wigler's group and Jonathan Sabat, that essentially, when you looked at individuals with developmental delay, autism, and later on diseases such as epilepsy and adult schizophrenia, there was an excess of large CNVs in patients when compared to controls. So what this means is that in principle, large swaths of the genome, when deleted or duplicated and found in excess or deficiency, were predisposing individuals to develop these diseases. Our own take on this began around 2002, and this is right at the kind of the end or the beginning, I guess, of the sequencing of the human genome, where we developed a map of areas of the genome that would be unstable as a result of their genomic architecture. So shown here is a map, and all the blue lines represent regions of the genome that have been historically copy number variable. So these represent segmental duplications in our genome. And back in 2002, we mapped out in gold all the areas of the genome that we thought would be prone to copy number variation because they were flanked by large, highly identical duplications. So pulling a page from the playbook of people like Jim Lupsky and others who had worked on unequal crossing over as a cause of developmental disorders, we predicted that these regions, because of the genomic architecture in the human species, would be particularly fragile and therefore prone to recurrently delete or duplicate every time we produced gametes as a result of unequal crossing over. So back then we identified about 111 such candidate regions, and at that time we knew that 23 of them were associated with developmental disorders, so particularly diseases associated with neurologic disease in children. And we targeted these regions using some technologies which are now, in some respects, out of date, to look for copy number changes in patients with unexplained developmental delay and autism spectrum disorder. I won't go through all the details other than to say that the approach was somewhat successful. We identified on the order of about a dozen new recurrent deletions. So sites of the genome, when deleted or duplicated, were found almost exclusively in children with developmental delay. So what I'm showing you here is a picture of children which is now called the 17q2131 deletion syndrome, also called the Kulin DeVries syndrome. So in 2006, we identified this portion of chromosome 17, about 500 kilobases in size, that was deleted in these children. So in a cohort of roughly 300 kids, we identified 4 children that had this particular segment of DNA, one of their 2 chromosomes, this 500 kilobase piece deleted. All of the cases were de novo. That means none were transmitted from a parent. So it had occurred presumably early in development or occurred during the production of the germ cells of the parents. And all the children had similar characteristic facial features as well as other aspects. You can see, I think, from this, you don't have to be a clinician or a dysmorphologist to appreciate the similarities in the way these kids look. They're all unrelated individuals. They all have this characteristic bulbous nose, pronounced philtrum, protruding tongue. And something you can kind of see from this picture is that these children have been generally regarded as children that have a behavioral disposition, that is, they're actually quite congenial and happy children. And so there's now been over on the order of 300 families that have been identified with this specific recurrent deletion, mediated by duplications that were a hotspot for copy number variation in our genome. Later in 2007, we identified this region. So this is a 3 megabase pair deletion occurring on chromosome 15. It takes out about a dozen genes in that region. And so in these children, they have one normal copy and then one deleted copy that they've inherited or they've actually received from one of their parental germlines. All of these cases were once again sporadic. That is to say, there is no transmission of event, but it's a de novo event in the germline of one of the parents. And these kids, once again, had characteristic facial features. You can see here the frontal bossing of the forehead, thought to be the result of an overgrowth of particular neuronal cells, the almond-shaped eyes. And what you can't see from this picture is that these kids are relatively short stature, so they're in the 25th centile worth of growth. This is the last one I'll show you. This is a deletion on chromosome 15q13.3. We documented this one in 2008. This one was different from the other two in the sense that not all cases were de novo. That is to say, not all were sporadic. Some cases, about 50% of the time, the events were inherited, often from a mother. 50% of the time, they were actually de novo. That is to say, they were not found in any of the parents. This is a deletion of about 1.5 megabases. There's no characteristic facial features with these individuals. However, these kids, in addition to suffering from mild developmental delay, typically essentially have a susceptibility to seizures. And work that I won't go into, but work that we did with groups focused on epilepsy, this particular deletion was found to be associated with about 1% of individuals with idiopathic generalized epilepsy. So these approaches allowed us to kind of get a sense of the importance of copy number variation with respect to developmental delay, and then autism spectrum disorders more specifically. And what I'm showing you here now is essentially a kind of a burden analysis looking at the proportion of children that come into a clinic with a diagnosis of developmental delay compared to controls. So the way to read this particular plot is what's shown here on the x-axis is the proportion of individuals in the general population or in kids with developmental delay that have a large deletion or duplication greater than 500 kilobases in size. So you can see from this plot that about 25% of children that are referred to clinics for unexplained developmental delay have a large copy number variation compared to about 10% of the general population. This has been used to suggest that as much as 14% of the quote-unquote genetic cause of developmental delay can be explained by these large copy number variants. One of the interesting things which I'm often asked is that these individuals here, these particular events that you see, so 10% of the general population, which means 10% of the people in this room, have a large CNV. Those CNVs are almost never seen more than once or twice in a cohort of about 10,000 normal individuals. And the data suggests that those CNVs are under relatively strong selection as well, so they're being eliminated from the population. The reason we have such a high frequency collectively is because we have our genome is structured in such a way to produce these large copy number variants recurrently. But this would argue that even among the 10% of the general population that are carrying large CNVs, they're probably contributing at some level to essentially their health or essentially maybe their deficiencies with respect to health because they are selected against and never reach particularly high frequencies. But these data, I think, were important because they suggested to us that copy number, and in particular dosage imbalance of genes, must be a particularly important mechanism by which we have children that have developmental delay or autism. Just to break this out a little bit more by disease, so if we take all of those patients that we've looked at, these are all children with various forms of developmental delay and various diagnoses, you can see that the burden, depending upon disease, is different. So this is the same plot I showed you before, but now breaking out children with craniofacial abnormalities, children with cardiovascular, so this is largely congenital heart defect, versus individuals with autism and epilepsy, compared to children with pediatric asthma. You can see big differences in terms of the burden of CNVs. You see for diseases like congenital heart defects, like craniofacial abnormalities, that we see the highest burden of large CNVs. Diseases like autism and epilepsy are intermediate. And diseases like pediatric asthma, we see no difference in terms of the burden. So it's not as if every pediatric disease is essentially impinged upon by large CNVs, but a subset, particularly those related to development and in particular development of the heart, development of craniofacial, development of the brain. We've also had the opportunity to look at inheritance of these large CNVs. And so this is data from about 2,500 families where we've actually looked at large CNVs and looked at what proportion are actually inherited, and if they are inherited, are they inherited from mother or father, and what proportion are de novo. And I've specifically broken out both the deletions and the duplications so you can see the difference. So you can look at this and you can say, well, wait, there's a big difference here between deletions and duplications. A much larger fraction of the deletions are actually de novo, and this fraction gets greater for both deletions and duplications as a function of size. And this should make sense, right? Because larger events involve more genes. Deletions are generally thought, based on most organismal studies, to be more deleterious than duplications, and therefore much more likely to arise essentially almost exclusively by de novo events. So when you get out to this range of 2 megabases, the vast majority of events that you see are de novo. Now, if you have a keen eye, you also see that there's a difference between the male and the female contribution. So if you look at what's contributed from the mother and the father, you'll see that the area under the curve, at least for essentially female transmission, is greater. And this is something that's borne out now over and over again when we look at severe events, not just large CNVs, that 58% of these essentially private large events, when they are transmitted, are coming from mothers as opposed to fathers. Now, some of you might be sitting there going, well, 58% doesn't sound like a large number or big difference as opposed to the expected 50%. But the number of cases and number of transmissions that we've looked at here means that this is a highly significant observation. So when transmitted, mothers are more likely to transmit or be carriers of events. And then some recent data, which I'm not going to go into, suggests that, in fact, sons, as opposed to daughters, may be preferentially obtaining these CNVs, at least in terms of manifesting a disease state. So we built our initial maps of copy number variation, studying really on the order of 10,000 cases and 15,000 controls. We've now pushed this in the last year to really look at it on the order of about 30,000 cases with developmental delay and 20,000 controls. And what we've been able to do is essentially build a morbidity map of human copy number variation, at least for pediatric diseases such as developmental delay and autism spectrum, based on the pattern of CNVs. So areas where we see an excess of deletions shown here in red or duplications shown here in blue in children that are affected compared to controls gives us some indication that we've actually identified a region of the genome when either deleted or duplicated results in an increased risk of developmental delay. And just to give you some perspective of what I talked about a few minutes ago, these hotspot regions in yellow flanked by segmental duplications are shown here. And when you get to these large numbers of 30,000 cases versus 20,000 controls, you begin to see patterns emerging on specific chromosomes where we start to see excess of events outside of the hotspot regions, which for us was particularly exciting because we now have kind of genome-wide ascertainment, even though it's just nominally significant in terms of excess deletion, excess duplication in cases when compared to controls. So this new morbidity map allowed us to identify 70 regions of the genome, many of which we knew already, but 14 which were newly significant. So it's important to continue on this work. I think a large perspective really from that of really the clinician and people that are doing diagnosis. We continue to identify new copy number variants associated in patients that we've never seen before. And if we don't pool large numbers of data together, we won't be able to provide any really risk assessment for families and patients that actually have these particular CNVs, because there is a background of really benign or at least subclinical CNVs within our genome. But probably the most important thing that came from studying CNVs for about 6 or 7 years was essentially a model for thinking about the genetic architecture of diseases such as developmental delay or autism. The first thing that we learned was that dosage and balance is very important. So having too much or not enough of a given gene, probably early in development, is actually critical. If you think about it, we kind of knew this from diseases like Down syndrome and some of the syndromic disorders that were described for CNVs early on, but this, I think, really reinforced it. The other important point was the model here is heterozygous. So we're talking about events where there's still a normal copy left in the genome, but the mutation is primarily a dominant one, at least in terms of our relatively simplistic thinking on this. The other important point, this gets to the question of common variants versus rare variants. At least for these diseases, each of these individual CNVs is rare. So affecting 0.5%, 0.2%, 0.1% of kids with developmental delay or autism spectrum disorder, but collectively quite common. So we're talking about 15% of developmental delay for CNVs greater than 500 kilobases in size, and that doesn't get close to the smaller events. So that's a minimum of its impact. For autism, 7 to 8%, in terms of its impact. The third lesson, or fourth lesson here, was essentially genome first. In many cases, we took things like broadly defined developmental delay, we found CNVs, we found recurrent CNVs, and then we studied the phenotype of those patients to identify new syndromes that had been previously unrecognized. So this idea of going from a genotype, which is in this case a CNV, to essentially a phenotype, was something that really emerged from the studies of CNVs. So CNVs have been fantastic, and I think are one of the biggest things that came out of the Human Genome Project, because it allowed us to explain to a lot of families the cause of developmental delay or autism in their specific cases. But they have actually sucked from one very specific perspective, is that most of the events that are thought to be pathogenic are large. And so they typically affect multiple genes, usually a half a dozen to a dozen genes. And in most cases, the actual individual gene responsible for the dosage imbalance and therefore disease is not known. So this is where I think exome sequencing came in to completely kind of fit the bill. So the idea that you could actually go back and do the same experiment, but now looking for, really with microscopic and submicroscopic precision, look for small events that actually disrupt genes. So all of you have heard of exome sequencing, this idea, it's kind of poor man's sequencing. Instead of actually sequencing an entire genome, what if you just go back and sequence the parts that we can functionally interpret, i.e. the protein coding portions? So that reduces the search space from really 100% of the genome to about 1.5% of the genome as a target. So if CNVs provide us this kind of 30,000-foot view of the landscape or genetic architecture of diseases like autism and developmental delay, exome sequencing would be kind of the bottom up. Go back to those families where we found no large CNVs, start to sequence the mother, the child, and the affected individual in that family, and try to look for the same type of mutations, gene-killing mutations that would have the same effect as a large deletion event, i.e. take out a gene almost in its entirety. So this was the model. So for the rest of the talk, I'm going to talk about the work that we've done in terms of sequencing autism exomes. And so the hypothesis was to essentially look at families, and particularly look at families where there's no family history of autism, which turns out to be roughly around 70%. There are families that do have autism history, but for the most part, most are sporadic or simplex, also called out-of-the-blue autism, so one affected individual. Sequence the mother, father, the affected child with autism, and if possible, and if money's around, sequence the unaffected sibling as well. With one simple idea. The idea would be to compare the de novo mutations between this individual and this individual. And obviously you need the parents for that, so find the sporadic mutations, and ask the question, is there a difference in the significance in terms of the impact of the mutations that you see in this affected individual compared to the unaffected? In other words, is there an excess of loss of function or gene disruptive mutations in the child with autism compared to their unaffected sibling? So 2012 was a banner year, I would argue, for sequencing autism exomes. There were 650 families that were sequenced in 4 studies. Ours was one of them. We targeted about 250 families in our study. We sequenced the exomes to what we consider pretty high coverage, about 80-fold sequence coverage. We got about 92% of the protein coding portions. And in a subset of the 600 families, a little over 350, both an affected as well as an unaffected individual from the family was sequenced in addition to the parent. So some of my students and postdocs would kill me for summarizing all this work with 2 bullet points, but here it goes. There was good news, and the good news was this, was that when you looked at the de novo mutations in this individual affected with autism compared to the unaffected, there was a 2-fold excess of gene-killing mutations compared to the unaffected. The overall rate of new mutations was not different, so there were about the same number of new mutations, but the type of mutations was different. So these individuals were twice as likely to have a gene-killing new mutation compared to these. This is important because it actually suggested that the model of CNVs, which was large deletions, could be applied at the level of de novo mutations for frame shifts, indels, and splice site, splice donor mutations. The bad news is when those 4 papers came out, and everybody gathered together and compared notes and said, okay, which genes are the autism genes? The bad news is that we never saw lightning strike twice at the same gene. So only 18 genes were hit recurrently, And that is actually significant. You wouldn't expect to see that many, but we did see 18. And in terms of loss of function, only 5 genes were seen with a recurrent loss of function. So independent gene-killing mutations in independent families. So back of the envelope calculations suggested that there were 500 to 1,000 independent genes, when mutated with a loss of function mutation in a simplex model, could result in autism. Why was that bad news? The bad news is that we couldn't prove pathogenicity for any one of these genes yet, so we couldn't tell anything to families really or tell anything to individual clinicians. And the bad news for a clinician would be that if you've been studying autism for the last 30 years, the chances that you've seen the same genetic cause more than twice in 30 years of practice is almost zero. So every child with autism is truly unique in the perspective that a different mutation in a different gene is going to be responsible for autism. So that was a bit of a sobering moment for us in the field, because we realized that this problem was, we had appreciated there would be locus heterogeneity. I guess we just didn't appreciate there would be this extensive in terms of the numbers. In our own study, we uncovered a few other things I just want to mention. One of the things that we had the ability to do when we started cataloging more than 250 new mutations, a small subset, about 20% of them, we could determine in whose germline the event likely occurred. And the reason we could do that is we would have a new mutation in the child, and then we would have in close proximity a SNP that was informative, so we could say whether it came from the mom or the dad. So when we did this analysis back in 2012, what we found is that 80% of the new mutations were coming from the dad's germline. In a study that was published about 3 months later by Kari Stephenson looking at full genome data, the data was essentially identical. About 80% of the new mutations genome-wide come from the father's germline. Moreover, if we looked at our patients that actually had 3 or more new mutations in their exome 2, 1, or 0, and we compared it as a function of the paternal age at the time of conception, you see this very modest but very significant correlation with advanced paternal age. In other words, older fathers are contributing more new mutations, probably through their germline production of sperm, to their children as opposed to younger mutations. This fits very nicely with the epidemiological data. Beautiful studies that have come out of Sweden have suggested that the risk of having a child with autism from a 50-year-old father is about twice that what it is for a 25-year-old father. This is not a property just of autism. This is a property of biology. Old men contribute more new mutations to their offspring. The other thing, which I have to totally be confessed, I was a bit skeptical of, is my postdoc was extremely disappointed that he didn't see a lot of recurrences. So what do you do when you don't see recurrences? You start to look at the genes in networks and in pathways to see if there's anything common about them. So Brian O'Rourke looked at what he classified his most severe mutations, likely gene disruptive mutations that he saw in the initial 200 plus families that we looked at, and severe missense shown here in black. So the color are likely gene disruptive mutations, and the black represents severe missense. And what he found was that when he looked at the protein-protein interactions among these, he found that 39% of his severe mutations were part of one highly interconnected protein-protein interaction network. I was skeptical, and I'll tell you why I was skeptical, because I feel that the databases are largely incomplete in terms of protein-protein interactions. There's lots of false negatives, there's lots of false positives. And so Brian had to go through a number of hoops, and in part coming from reviewers' comments. So he showed that this was actually statistically significant based on modeling. He went ahead and actually showed that if you actually looked at what was defined as autism genes, this network, which he called a beta-catenin CHD8 network, was highly enriched. And the only part that convinced a reviewer was essentially going back and looking at the pattern of de novo mutations in the unaffected sibling and basically not seeing this network in those unaffected siblings. So he reported this in 2012 as a CHD8 beta-catenin protein-protein interaction network, largely because of the hub position of both this chromodomain helicase CHD8, which we had found 2 recurrent loss of functions in terms of our autism families, and beta-catenin, where we had seen some severe de novo missense mutations. Other work coming out from other labs, particularly from Ivan Iosifov's lab, also found an enrichment for genes that interact with FMRP, or the Fragile X mental retardation protein. So there seemed to be some logic to the types of mutations that we were seeing in the autism individuals, and enrichments, particularly in certain pathways. So fast forward now 2 years. The 3 of the 4 groups that had actually competed with one another actually joined forces to actually publish one kind of comprehensive assessment of 2,517 families, about 10,000 exomes that were sequenced between Matt State, Mike Wigler, and our group. Of these, there were 1,900 quads, so that means there was one unaffected sibling, and there were 606 trios, so that means there wasn't an unaffected sibling, just an affected individual. A total of 2,162 validated de novo mutations, and all the recurrent and all the truncating mutations were validated by Sanger sequencing. So as of end of last month, or I guess the end of December, we had 345 genes which had 1 or more de novo truncating mutations based on this analysis. 70 genes have 1 truncating mutation and an additional de novo missense mutation. 26 genes essentially with multiple loss of function de novo mutations never found in siblings or in controls. The estimate on these 26 genes is about the probability that they actually are contributing to risk or significant risk for autism is about close to 95%. We've also identified a large number of genes with 2 or more de novo missense. There's 83 genes in 178 patients, and there's some that have 3 or more, 12 genes in particular, giving us roughly, again, back of the envelope, about 200 high likelihood, I would argue, candidate genes for de novo mutation associated with autism. We revisited the story about the excess of de novo disruptive mutations, and the result holds. So this is data for nonsense mutations, splice site mutations, and this is the collectively likely gene disruptive mutations. This is the total number of events that you're seeing in red in probands compared to the unaffected siblings. So you can see this pretty much this consistent 2-to-1 pattern in terms of probands having more. You actually can calculate the number of events per child. So these are the number of mutation events de novo per exome for truncating events, and you find there's 0.21 in probands versus 0.12 in sibling, resulting in a differential of about 0.09 over 0.21, allowing us to estimate that 42% of the truncating de novo mutations are contributing to autism spectrum disorder. In other words, when you sequence a family with autism, with simplex autism, there's almost a 50% chance that if you see a de novo truncating mutation, that de novo truncating mutation is contributing to autism. The really cool thing, which was subtle but important, was that when you look at missense mutations for the first time, because we had the large numbers, we now saw significance. So we see an excess of missense mutations in probands, de novo missense, compared to unaffected siblings. Note that the number of synonymous mutations is about identical between the two. So we can do the same calculation and estimate that 12% of the de novo missense mutations, based on this differential, are contributing to autism spectrum disorder. That means that 9 times out of 10, when you find a de novo mutation in a child with autism where there's no family history, it has nothing to do with autism, but 1 times out of 10, it does. Interestingly, when you ask the question of how much does each of these contribute to the overall kind of risk of autism, these are about equal numbers. That's because there's so much more de novo missense mutation. So combined, roughly 20% of autism may be accounted for by de novo missense, likely severe mutations, and de novo, likely gene disruptive mutations. One other point I'll make, which echoes back to the CNV work, we did a fairly extensive search for all the events that we saw, including the recurrent ones for loss of function. And we found no evidence of compound heterozygotes. This would argue pretty strongly that it's not a recessive model, but just like the CNVs, probably a dominant model with respect to autism. So for those of you interested in genes and gene lists, these are the top hitting genes. CHD8 right at the top, 7 cases, nothing in missense. There's not a single case in the ESP, or in fact in terms of over 6,000 controls have been sequenced in terms of loss of function mutations. Recurrent missense mutations, some very interesting genes, such as a sodium channel gene. This is a master transcriptional regulator, CREB-BP, where we're seeing essentially excess of de novo missense mutations. But this is the problem, locus heterogeneity. 500 to 1,000 genes just in a simplex model. How are we ever going to prove any of these at an individual locus level? So thankfully, I was in a department where a person by the name of Jay Shinduri was working on new technology to solve this problem. It's called one of many different approaches, but this one is called molecular inversion, probe technology. He developed it in part in George Church's lab and then really perfected it when he came to Seattle. And the technology is conceptually very simple. Essentially, it's glorified oligonucleotides with complementary arms to your target, let's say exonic regions that you want to, of genes you want to resequence. You design thousands of these corresponding to the genes you want to resequence. You put them in with your DNA from your individual DNA samples. You then throw in a DNA ligase as well as a polymerase, and you form a covalently closed circle. The exonuclease treat to actually remove single-stranded DNA, throw in a barcode, PCR amplify. You then pool all these DNA samples, and you can pool 192 or 384 and run in a single lane of Illumina sequencing. The important point of this technology is that it's rapid, sensitive, very specific, allowing us to sequence essentially 192 samples or 192 individuals for 50 genes, covering about 92% of the target for about $12 a sample. So this is exactly what the doctor ordered, because in order to prove any individual gene, you now have to resequence it not in hundreds, not in thousands, but ultimately tens of thousands of samples. And this technology allows us to actually do that. So if exome sequencing is poor man's sequence, this is poor man's sequence in terms of actually getting at these specific genes. So this is what we did. So we did a pilot in 2012. We targeted basically 50 genes that were our best candidates. And what we found was essentially the following. When we targeted essentially individuals with autism, so this is roughly about 2,500 individuals that were sequenced, we found 8 de novo mutations in the gene CHD8. 7 of them were loss of function mutations. None were seen in over 6,000 controls that had been exome sequenced. GRIN2B, which is a glutamate receptor, we found 4 de novo mutations. 3 of them were loss of function mutations. None had been observed loss of function mutations in controls. DYRK1A, a dual tyrosine kinase, this one is actually 3 out of 3, so 3 de novo loss of function mutations, never seen in controls. And TBR1, which is actually a marker of cortical development, it's actually 3 out of 3 loss of function mutations. So we can actually compute and build a model to estimate what is the likelihood that you would expect to see 3 or 4 or 2 de novo mutations that will be likely gene disruptive. And based on distance and the size of the gene and mutation rate of a locus between chimp and human, we can do that. And so in this model, what we've done is actually computed the likelihood that you would expect to have seen that many de novo mutations that it would actually be gene disruptive. And when we did this calculation, 5 of the genes that we originally targeted actually reached locus-specific significance. So now instead of saying we have an aggregate signal, we now can say specific genes like CHD8, like GRIN2B, like DYRK1A, when you see a loss of function in those particular genes de novo, that they're likely contributing to autism. And while each of these contributes to a small fraction of the total, collectively these 5 genes account for 1%, or just a little bit under 1% of autism. So this is just the beginning. But the really cool thing is when you start to get to these numbers of having more than 2 or 3 patients that actually have a given particular mutation, you can ask simple questions. Is there anything more similar about those patients than, let's say, your generic autism from which these patients were drawn? And so I'm showing you data here from essentially head circumference data. So some of you may know that children with autism have generally been described as actually having larger heads. And that's true for a subset. It's actually a little bit of an oversimplification. This is the head circumference distribution. This is the normal in the dotted line. And you can see that the actual head circumference data, the variance is actually wider. So you have some individuals that have larger heads, some of the individuals that have smaller heads. But if you actually map onto the actual patients that have CHD8 loss of function mutation, you can see that 6 out of 8 of those patients would classify as actually macrocephalic. 2 of 3 of the actual DYRK1A patients would classify as microcephalic. So why am I telling you this? Well, we know that when you mutate DYRK1A in Drosophila or mouse, it actually results in a reduction in brain volume. This gene has been studied for many years because it's thought to be one of 2 genes important for the cognitive deficits associated with Down syndrome. Now, Down syndrome is a gain of an extra copy. This is a loss. So people have mutated this and showed quite convincingly in a number of different studies that you have a reduction in brain volume. And the data would seem to recapitulate that in humans. CHD8 not much was known about it. Still, I would say it's still largely a black box. It's a chromodomain helicase. It's a regulator of chromatin. It's thought to negatively regulate beta-catenin. Beta-catenin is expressed everywhere, it seems, and it's important in many different processes. But in work done by Chris Walsh, if you constitutively express beta-catenin, you result in overgrowth of mouse brains. So mouse brains will actually develop gyrifications if you actually constitutively express beta-catenin. So if you have a negative regulator of beta-catenin, you might expect essentially a macrocephalic phenotype if you actually knock that out. So the question is, do we see, for example, mutations in beta-catenin? And the answer is yes. We now have identified 3 mutations in beta-catenin, severe mutations that are de novo, and these patients place on the exact same place where you'd expect kind of the opposite of the negative regulator of CHD8. This is just to show you the data in the context of the family. So we have the probands here with DYRK1A mutations in blue, CHD8 patients in red. So these are de novo loss of function mutations compared to their parent, sex-matched parent versus their sibling. And I think you can clearly see the trend. The DYRK1A finding is significant, and the CHD8 is actually borderline significant. So encouraged by this, we actually set up to then kind of expand the search. So instead of doing a few thousand, let's do 20,000. So we now have a list of about 245 genes that we want to interrogate systematically. We've put together a collection of 6,000 autism, 6,000 children with intellectual disability / developmental delay, and 6,000 controls. And we're systematically going in and we're resequencing these individuals, searching for recurrences, trying to find evidence that we've actually have identified, first, a pathogenic locus relevant to autism or to developmental delay. And then second, if we actually find them, let's say 5, 6, 7 patients, can we reassess them for phenotypic similarities? So we now have a cohort, I call it the Sid Network, of roughly around 20,000 patients. It's a little bit less than that right now. It involved essentially 10 out of 13 of the original participants in the Simon Simplex collection, and another 10 labs scattered across 4 continents where patients have been collected, DNA is assembled, and we are actually sequencing it with IRB approval. And that was no small task, let me tell you, because in these particular study, we actually had to break the code for the individual patients and go back to each of the individual sites to actually get IRB consent to do recontact. And then if patients are found with a particular molecular lesion that we think is pathogenic, we then bring those patients back in for clinical reevaluation. That's the fundamental distinction, I think, from many of the collections that are at NIH, is that we actually have phenotypic recontact a possibility for most of these samples. So these are those, for those of you interested, we have collaborations from all around the world, a lot from Europe in particular, and these are the results. So the first results came in really the summer of last year. We actually went after CHD8 because that was the one that we had the most hits. We now have 20 truncating mutations in CHD8. We reported 13. Of these, essentially 11 of the 12 are de novo, and 0 out of 8,792 controls have actually shown a truncating mutations. So this is probably our most penetrant and most significant gene for autism that have been identified to date, at least for de novo mutations. It's, again, this chromatin helicase DNA binding protein, thought to be a negative regulator of Wnt signaling and beta-catenin. In a model put forward by Nishiyama, it's thought that this actually affects many genes during development. And the way it does this is that beta-catenin interacts, or CHD interacts with beta-catenin, recruiting H1 to essentially create chromatin compaction and then for transcriptional silencing at specific sites during development. What do the patients look like? So we've actually been able to bring them in. Many of them have come back into Seattle and have been seen at the Autism Center, where they undergo a 2-day evaluation involving everything from a reevaluation of the diagnosis of autism to essentially a meeting with pediatricians, doing MRI if that's possible, as well as other workups. So of the patients that we've been able to ascertain, 87% of them have a clinically defined definition of autism. And remember, we're pulling from both developmental delay and autism when we're doing this study. So this is actually a good sign. This would suggest that this is the primary diagnosis here would probably be autism. Less than half of these individuals have intellectual disability, so it's not primarily an intellectual disability gene. 80% have macrocephaly, so this is in fact predominant and early during development. So we see this in the first month to 2 months of life based on longitudinal medical records. These kids are at the 95th percentile pretty much after the first month of life. One thing we hadn't appreciated was that 73% of these patients have severe gastrointestinal dysfunction. This is a significant finding compared to the Simon simplex. This is typified in these families by bouts of diarrhea, often weeks on, followed by bouts of constipation, which seems to last until puberty, in which point it somehow, in many of these families, has ameliorated, at least on some of the follow-ups that we've done. One thing that isn't significant is this one, but we think it's an interesting observation. Many of the families are reporting severe sleep dysfunction in their children. The first 2 families that came in, for example, reported that their kids would stay up sometimes 3 days consecutively without sleeping a wink. To one point that one mother even commented to us that they had to actually lock the doors at night because they could not stay awake and they were afraid that the child would wander off because the child would never sleep and then would suddenly power nap for a long period of time. We'll need larger numbers to see if this will hold up, but I think it's interesting because this essentially pursuit of the individual gene to the phenotype is actually revealing to us new features of at least this particular form of autism. We've made some attempts to model this in zebrafish, and there's actually experiments ongoing to do this in mouse. So this is work of postdoc Bo Zhang, just looking at expression of this CHD8 gene in zebrafish. You can see it's expressed early on, and then it becomes more restricted to the head region later during development. And if you can see here, it also becomes pretty intense along the gastrointestinal tract. So you see this black line indicating that it's expressed there. In morpholino experiments, followed by some CRISPR work, we've been able to show that, in fact, in developing zebrafish, that there's an increase in intercusp distance from 4 different morpholinos, as well as a CRISPR knockout, consistent with the overgrowth that we see. Marker analysis, which I'm not going to show you, suggests that particularly dorsal progenitor neurons, there's an overproliferation of them in the developing zebrafish. And some interesting work from Christel Goltzio in the same zebrafish. What they've done is looked at this gastrointestinal dysfunction in an assay that's called a microgavage assay. And what you're doing here with these morpholino fish is you're essentially stuffing fluorescent microspheres into their mouth, and then over, as a function of time, measuring how far they traverse along the digestive tract. So dividing it up into zones 1, 2, 3, and 4. So when the microspheres are completely expelled, they will come out over here at the end. So these are control fish, and you can see at 3 hours post-injection, a significant fraction are in zone 4, and by 6 hours, most are expelled. But in the morpholino fish, as well as the CRISPR fish, they seem to get hung up. So they get stuck in zone 3 and 4. Even 6 hours post-injection, they still haven't completely expelled. This is a significant finding, and it would suggest, for example, that this may be mimicking some of the effects that we see in patients. Interestingly, and I don't have a completely good explanation for this, if you stain for enteric neurons with HUCD antibody, this is a normal fish, this is a morpholino fish, there's a consistent depletion of enteric neurons, suggesting either a migration or a proliferation defect. But if you remember, this is the exact opposite of what we've seen in developing forebrain or the midbrain. We see an overproliferation of neuronal precursor cells. This is another subtype of autism that we've been recently kind of characterizing with our colleagues in Belgium. This is essentially a gene known as ADNP. We now have 10 loss of function mutations in patients versus 1 out of 8,000 controls. 9 out of 9 of these that we've seen in patients are actually de novo. So that's to say that they're not inherited from the parent. You can see here similarities in the phenotype. So these kids all tend to have essentially this kind of mild hypertelorism. But you'll also notice this high anterior hairline, this frontal bossing. And the data from some of the MRIs suggests that, in fact, there's an overgrowth, particularly of the neocortex in these particular children. The rest of their brain seems particularly normal. 100% of these kids have intellectual disability, 100% have autism, and 90% have almost complete loss of expressive language during development. So it's a pretty severe form of autism, even from the perspective of language development. We don't have to make a mouse knockout. It's already been made for a completely different reason. This gene, activity-dependent neuroprotective peptide, has apparently been studied for many years as a potential therapy for Alzheimer's. It's a presumed transcription factor, once again associating with SWI/SNF chromatin remodeling complexes, important in cell proliferation, thought to be important in tumorigenesis. ADMP knockouts, this is homozygous, essentially have failure for the neural tube to close and therefore die. ADMP hets, or heterozygous mice, actually have a neuronal glial pathology and a reduced cognitive functions. So this is a particularly interesting case. We really think this is identify what is previously thought as idiopathic autism, but now defining a very specific syndromic form of autism. Last example I'll show is this dual tyrosine phosphoregulated kinase. This is essentially the Down syndrome cognitive dysfunction gene, now deleted. In a paper that will come out at the end of this month, we've actually characterized 8 truncating mutations from 7,000 cases and just found this loss of function in none of the controls that have been sequenced or exome sequenced at this point. 7 out of 8 are de novo. And the kids actually are quite interesting. You might be able to guess which kids actually were diagnosed with developmental delay and which ones were diagnosed with autism. All the autism kids are along the top. All the kids with developmental delay are at the bottom. There's obviously great range of phenotype here. 100% intellectual disability, 100% microcephaly. So now that's pretty much well established. 89% of the patients have late onset epilepsy, as well as a definition of autism for 83%. They have these typical facial features, which you can see here. It's kind of this reduced jaw in particular, this relatively pointed nose that has been observed by many of the clinicians. This is actually pretty severe in these patients because some of them have actually had reconstructive surgery to accommodate the eruptions of teeth as they actually get older. We can't explain the heterogeneity in this one. This one is not so straightforward, but it's pretty clear that this is a microcephalic, so reduced brain volume. And this is supported by data, as I mentioned, from mice as well as from Drosophila. Drosophila mutants result in a phenotype called mini brain. And in mice that are heterozygous, you can see reduction in the mesencephalic tectum, medulla oblongata. And heterozygous mice have problems with their reflexes, particularly writing reflexes and eyelid opening and various other classes of behavioral, pre-weaning behavior. So we're going forward and doing many more genes. We now have significance for new genes that have previously been unrecognized, such as a gene known as TRP12, which is a thyroid interacting protein. It's also an E3 ubiquitin ligase. PAX5, which is a paired rule gene, important in terms of development. But the important point here is that I think genome sequencing, and in this particular case, exome sequencing, has actually provided us a model for going after a significant chunk of the genetic etiology. And by virtue of the fact that we can go back in and restudy those individuals with a common genetic etiology, we can begin to break down the umbrella of autism really into maybe many subtypes. So in summary, I'll conclude with a couple comments. So the beginning part, I talked about CNVs. And CNVs were great, I think, for making us rethink as geneticists how we should approach the study of developmental delay in autism. Instead of it being an inherited disease, we're thinking now in terms of actually more of a sporadic disease, perhaps with private mutations playing an important role as well. Exome sequencing data has actually provided, I think, a lot of insight in terms of specific targets. So 42% of de novo loss of function we now believe are contributing to autism spectrum disorder. 12 to 13% of de novo missense mutation, combined with CNVs, sporadic missense, sporadic loss of function. We now think we have a handle on 30% of the genetic, and I'm going to be careful here, put in quotes again, cause of autism. But we haven't proven the case for more than a few percentage at this point. So I think that's an important thing to distinguish. I love the idea of a genotype first based approach. This is proposed not first by me, but by others, including Les Biesecker here at NIH. This idea that you actually start with the genetics. You let the genetics to sort out common or individuals with a common genetic etiology, and then you study those individuals more carefully to understand, in fact, whether in fact there is subtleties about the phenotype that have been missed, at least in terms of a generic diagnosis such as autism. So if I can just basically give you my perspective of where we are in the field, this is definitely the glass is half full perspective as opposed to half empty. I think we've come a long ways since 2007, where the vast majority of the genetic etiology of autism was unexplained. We now have a handle, maybe 35%, maybe 40%, of where that missing quote-unquote heritability exists. I think the big unanswered question is what role does essentially the non-coding portions of the genome play? And the ability to now go back and look at the full genomes of individuals where we haven't found protein coding mutations and look specifically at genes that we know are dosage sensitive because we now have proven that they are in fact relevant, provides us with a logic, I think, for beginning to tease apart as opposed to just blindly sequencing genomes. This model of a genotype first approach I don't think is specific to autism or developmental delay. I think it actually be applied much more broadly. To me, it makes a lot of sense, but I have to confess I'm biased because I'm a geneticist. Start with genetics. Let the genetics sort out that you have a significant or pathogenic risk by finding recurrences, and then study the phenotypes of those individuals to ask, in fact, if there are commonalities between them. And in some cases there will be, in some cases there won't be. The perfect world would be one gene, you see recurrences with one phenotype, and then you basically know that you have a correspondence one-to-one. I think much more likely will be many genes will converge on, for example, macrocephalic autism, and they will actually maybe, in the end, there'll be some genes in which, in fact, depending upon where the mutation is found, the type of mutation may result in many different clinical outcomes. And maybe we're seeing that with DYRK1A. The important point here is that I think this is the first step for laying down which patients should be treated by which therapies. I think the idea that you would treat a child with autism that has microcephaly the same way you would treat a child with macrocephaly is absolutely ludicrous. If there ever was a therapy, they are actually counter-opposing, right, each other. And you would want to make sure you actually identify these distinctions early on at a molecular level. Last, the genes do make sense in some respects. They are falling into pathways and clusters that belong to specific processes molecularly. And so while 500 to 1,000 different genes sounds almost impossible to really tackle, the fact that they boil down to perhaps a dozen or so pathways I think is very encouraging. So we're seeing things, for example, like what I'm showing in this particular slide is essentially the relative size of the circle indicates the number of de novo mutations, red being loss of function, green representing here, or blue representing missense. You can see that these genes are part of protein-protein interaction networks and are highly connected, and they center around themes such as synapse plasticity and function, Wnt signaling, and chromatin remodeling. And so this is our kind of our core, if you will. If you just add all the loss of function mutations, about 60% of them can be mapped with one step or so removed from this PPI network. So my postdoc has given me an education. He's taught me that there is quite a bit of value to actually thinking about this disease from the perspective of pathways and networks, as opposed to just simply thinking about the genetics. And lastly, for those of you who actually, like myself, have family members, this is the hope that we have with studying autism. Genetics is really just the beginning. It's going to get us to genes, and once we prove those genes when mutated result in disease, we have something to offer to families. It's not a lot, but it's diagnostics. This is how it begins, and I think once we identify the diagnostics, smarter people will begin to work out the biology, the physiology, the biochemistry of these genes, the neurobiology of them. Ultimately, these will feed into networks, collections of genes that are connected both in time and space and perhaps in terms of function. And I believe when there are therapeutics, which isn't today, but hopefully in the future, that it'll be directed toward the networks. And so some people often ask me, why does it matter whether my child has type 1, type 2, type 3 genetically? And I think the answer is this. I think once you know why your child has autism, you will know what network ultimately they belong to, their protein-protein interaction networks. And if there is a therapy, I think the therapies will be applied to those subsets of patients that have a specific genetic lesion that makes molecular sense. So this is kind of the model of precision medicine that I think we're dreaming of, and I think it could be real for things such as autism, but it starts by really working out the genetics first and foremost. So thanks for your attention. These are the folks that really did all the work. Brad Coe for all the CNV work in particular. A postdoc is on the job market. Brian O'Rourke has already landed his job. He was a postdoc that worked in my lab and Jay's lab. Jay Shinduri for this excellent collegiality and work with the molecular inversion probe assays. Some other folks that have collaborated a lot over the years, in particular Lisa Schaefer, when Signature still existed. Collaborators, particularly from other labs, as well as in the clinical side, as well as the Simon Simplex Consortium. And last but not least, we couldn't do this work without the support of families and the patients. And so we're always indebted to the work, to their contribution, and the parents have actually been fantastic in terms of recontact and really working together to try to work out why their child has the autism or the developmental delay. Thanks for your attention. Happy to take questions. Yep.
Yeah, you mentioned with GI associations with one of the forms of autism. I'm wondering, has the term Hirschsprung's disease ever come up in terms of those cases? You weren't very specific, but Hirschsprung's would be a good candidate to think about, wouldn't you think?
Absolutely. We've done some comparisons on Hirschsprung disease patients with Aravinda Chakravarti.
I'm sorry?
Aravinda Chakravarti at Johns Hopkins has done work on Hirschsprung's disease, and we've been looking not at exome sequence data, but actually CNV patterns within those patients. And we find an excess of really CNVs, certain types of CNVs that we've seen in kids with developmental delay in Hirschsprung patients. The patients that I described here with the CHD8 do not have a diagnosis of Hirschsprung. But what you're suggesting is that there's a connection between neurodevelopmental and GI dysfunction is something I think that makes a lot of sense, and we've seen hints of this with Hirschsprung's disease already.
Okay, thank you.
Yep.
First, I want to thank you for your work. It's on a personal note. One question I have is, when you were talking about your pathways, has your pathways led to any methylation dysfunction in any of these children? And my second question is, do you have any population of the children that are not dysphoric, like they don't have any problems with their face or frontal bossing?
Yes. So the answer to the first question is, have we seen any methylation differences? We have not looked. I know others are looking in these same patients, but we have not compared notes. So we have not specifically looked in, for example, the patients with this chromatin helicase mutations, whether there's any, but it's a good suggestion. And in terms of patients that look normal, it's funny. If you would have grabbed the CHD8 patients, and we've tried this with some students that know nothing about the stories in our lab, and you took some CHD8 and ADNP patients, you just scrambled them with other autism patients. A clinician would never look at those and say, "Oh, that's a kid with craniofacial abnormalities." They never would. That's well within the normal range. The only reason you start to see it, at least that's the argument that we've been thinking about, because these are all described as idiopathic cases of autism, is that when you have 6 or 7 of them all around the same time, and you now study them carefully, you start to see these subtleties in their phenotypes. Now, the kids with DYRK1A, no problem. That's syndromic, at least half of them. But the kids with ADNP and CHD8 mutations, you wouldn't necessarily finger them as being essentially syndromic just by looking at them. So I don't know. I mean, when I look at the children, they all look, well, when I see the pictures of them, many of them seem quite normal looking to me, to be honest.
All right, thank you.
Yeah.
Thanks for a wonderful talk.
Thanks.
You were describing how these de novo events are sort of helping to identify territories within systems biology, I guess, networks that could be enriched for risk. Do you think there's a way of further parsing these networks by using the test of enrichment for association of more common variants?
Yes, I think it's a great idea, something I've been meaning to go back to. Now that we've resequenced a gene, let's say 8,000 times in cases versus controls, we have a whole panoply of additional mutations that we haven't bothered to study because they might be rare or they might be even common, but they're inherited, right? We've been transfixed on finding the de novo kind of mutations that are loss of function, right? But it'd be pretty straightforward, I think, now, because we've done the experiment, to go back to the data, reanalyze it, and look for essentially burden, which I think is what you're getting at. And for all the other cases where you haven't seen anything, maybe there's, in fact, some additional hits compounded with other hits in the genome that may be contributing.
Thank you.
I was wondering if you could specify what criteria was used to define a severe missense mutation.
Yeah, so that's a great one. So we've argued a lot about this in the lab in terms of what is severe and what's not. We've kind of settled on this. I'm not sure if you're familiar with this C score. It's also called the CAD score from Jay Shanduri's group. And so that one we used, I'm trying to remember what the definition was specifically in terms of the CAD score, but I think it was in the bottom, or the top, I should say, in terms of conservation, top 20, 30% of genes. The other metric that we've been using recently, we're just starting to publish on it, is this thing called the RVIS score by David Goldstein. So this is a measure of intolerance based on looking at data from the ESP or exome sequencing projects. And that actually seems to be pretty informative. And so we've seen signal at less than 50% RVIS and less than 20%. So really conserved genes are coming up in this screen.
Thank you.
Yeah.
Do you see any differences on how the patients get pregnant? In other words, are more of them in vitro fertilizations or anything else that can happen during the pregnancies?
Well, to be sure, most of the patients that we're studying, I mean, they don't get pregnant, right? Because they're children with autism, developmental delay.
No, I meant the parents.
You mean the parents?
Yeah.
Do we have data? I can tell you this, but I don't know what to make of it. For the CHD8 patients, what we've seen in those records, and I actually talked with the clinicians about this because I thought this may relate to the macrocephaly, is that something like 75% of the mothers actually had to have a C-section or had tremendous difficulties during birth. And this might be consistent with this overgrowth, right, which we see, at least in terms of longitudinal data, coming on very early during development.
But no particular increase, say, in vitro fertilization?
Not that I've seen. There's the odd record here and there of multiple miscarriages in the clinical reports, but there hasn't been a systematic survey of this.
Okay, well, if there are no other questions, I think we'll call it to a close, and we have the reception in the library immediately following.